Good morning to all. Myself Lalita. I'm working in Department of Microbiology. Today we are discussing about DNA replication and enzymes involved in DNA replication. DNA means it's a picture of showing a DNA. DNA is double helix structure. It's made up of adenine, guanine, cytosine, thymine, the four base pairs, and adenine always bind with diamine, and guanine bind with cytosine. The four base pairs are mainly found in DNA. The DNA is double helix structures made up of in phosphate group and sugar and nucleotide. These main base pairs of DNA. uh, DNA replication is nothing but it's a duplication, or sometimes called newly synthesized DNA is called DNA replication. Uh, DNA is it's semi-conservative method of DNA replication. Each new DNA molecules contain on one old strand and another one is newly synthesized DNA strand. The picture showing only newly synthesized DNA strand. This picture showing the model of DNA replications. The DNA double helix structures is unwinding the unwinding the polymerase and helicase and leading strand and lagging strand and RNA primers, template, SSP proteins. These are mainly founded in the DNA replications. The DNA difference between RNA. The DNA sugar molecule deoxyribonuclease and RNA is sugar is ribosomes present. DNA is double helix stranded molecules and RNA is single stranded molecules. RNA is only in thymine bind with adenine. Uracil RNA instead of uracil and diamonds. And then continuation, nuclear DNA is mitochondrial DNA and chlorophyll. These three main organelles of in present in DNA. And RNA mainly these types of mRNA, dRNA and RNA. mRNA means the messenger RNA, dRNA means transfer RNA, and rRNA means ribosomal RNA. They mainly founded in cell organelles. And then nuclear DNA never leaves on the nucleus and assembled in nucleus move to cytoplasms in the RNAs. The feature showing DNA ligase bind with phosphate groups and joining of DNA ligases and then cap shield with DNA molecules. This model of replication. The first is initiation. Initiation is first steps the DNA structures unwinding. Unwinding means helicase enzymes binded and then moving of polymerase three enzymes, the replications leading strand and lagging strand, and binding with the SSP proteins. Next, next step for elongation. Elongation means the additional of leading strand, lagging strand. Leading strand is nothing but continuous synthesis of DNA strand, and lagging strand it's a discontinue of synthesis DNA strand. It's a newly synthesized DNA strand 5 dash to 3 dash and 3 dash to 5 dash. It's a template of DNA strand, and then finally the new synthesized DNA strand. Third one, elongation and termination. Termination means it's a completely synthesis of DNA strand. The replication circle of DNA equally the fissure showing the circular DNA and the separate the positive coils. Elongation and elongation means unwinding the added some polymerase enzymes and copy of DNA, and the standard origin molecules is binded 5 dash to 3 dash and 3 dash to 5 dash. It's a newly synthesized DNA. It's bind with the SSP proteins and ribosomes formation of called Okazaki fragment. The Okazaki fragment is it's called lagging strand. It's newly initiated the Okazaki fragment and continually synthesis of DNA strand. DNA replication in eukaryotic. Eukaryotic is nothing but it's a chromosomal during S phase. It's a cell cycle of DNA. It's a synthesis of new DNA strand is called S phase. Next one, the cell large number of in the replications and the environmental favorable for the favorable condition of in DNA replication. The cell will not enter into the mitotic phase unless the DNA has replicated. The chromosomes also must be in attached mitotic spindle for mitotic or meiosis in the complete cell cycle of only in mitosis to the complete cell cycle. Next one, the CDK. CDK is nothing but cyclin dependent kinase. It's for the system of proteins induced in the cell cycle. Enzymes called cyclin dependent kinase. It's each chromosome have the double helix structure or on one linear strand. The template added with RNA primers and continually synthesized DNA. and synthesized DNA, newly synthesized DNA, new stand. The picture showing electron microscope of replications. For example, in Drosophila. What about the end of telomeres? Telomeres is the DNA is always 5 dash to 3 dash and 3 dash to 5 dash. It's a DNA polymerase ligase cannot fill the gap of in the end of chromosome, but the RNA primers is removed at the gap is not filled chromosomes. Okay, the shorter and rounder of replication solution. Eukaryote is randomly repeated sequencing end of the chromosomes. The telomeres composed with the protein RNA complementary to the telomeres repeated the terminal telomeres repeated and catalyze the addition of new repeats. Components of lengthening to the chromosomes or mutation or suddenly in changes absence of telomeres activity and result of chromosome shortening limited of cell divisions. The picture showing synthesis of telomeric DNA by the telomeres. The DNA sequencing is running. After that, the cap left, the primers removed, and telomerase enzymes added. The RNA of telomerase adding and filling of these enzymes, RNA primers subsequently removed, and then rejoining of DNA ligase enzymes. Final steps of assembly into the nucleosome. DNA is unwind nucleosome must be disassembled, and then histones are associated with the chromatin protein must be duplicated, newly synthesized protein synthesis. Especially in the replicated DNA and assembles nucleosome almost immediately. The histone is made up of protein H1, H2A and H2B and H3 and H4. It's supported to the DNA chromatin proteins. Once revised, the overview of DNA replication. DNA replication is duplication of or synthesis newly DNA molecules, and it's called DNA replication. There are two types. There are three types. One is semi-conservative and non-semi-conservative and discursive method. These three types of DNA replication, but DNA is mainly in synthesis of semi-conservative method of DNA replication. The replication, why is synthesis semi-conservative method? It's a DNA is double stranded and newly synthesized only new strand, another one is old strand. It's called semi-conservative of DNA replication. Okay, and then replication are three types are involved. One is initiation, another one is elongation and termination. These both types are in synthesis DNA replication. DNA is always bind with adenine, always bind with thymine, guanine always bind with cytosine. This main base pairs of DNA base pairs, and then nucleotide phosphate groups, sugar groups in attached with DNA backbones of molecules. The DNA, its double helix structure, its P form of DNA, it's discovered of Watson Crick. DNA is made up of proteins, nucleotides, and addition of diphosphate nucleotides. This is more in of the picture showing already. I told that DNA is double helix structure, unwinded, added DNA polymerase and helication unwinded, and the synthesis for newly DNA stand. This discontinually synthesis of DNA stand is called Okazaki fragment, and continually synthesis DNA stand is called leading stand. So it's a elongation process, and terminations finally synthesis DNA stand. It's added DNA ligase enzymes. Ligase enzymes nothing but it's a joining of DNA molecules. It's filling with phosphate groups and nucleotides. So it completely synthesis in the DNA stand. The application of genetic engineering using for cloning technology, and then recombinant DNA technologies. It's especially vector using insecting for gene cloning technology.